Good day, everyone. I am Angelica B. Morano, and for today's lesson, I will be discussing to you the introduction on the chapter of molecular properties of genes. So before we start, let us have first a short recap of the previous chapter that is discussed by group of Ms. Halop, Mr. Josue, Mr. Landesa, Ms. Lopez, Mr. Marquesa, and Ms. Mercader. So in the previous chapter, they discuss on the molecular structure and replication of the genetic material. So that includes the topics on the molecular structure of DNA and RNA, the chromosome organization and its molecular structure, now the DNA replication process, now the errors in the DNA replication, now its repair mechanism and recombination mechanism, and also the genetic code, the triplet code, the universality of the genetic code, and also the gene expression where dito ara discuss the transcription and the translation of RNA. So now in the new chapter in this um, lesson that we will discuss right now, we'll focus on the molecular properties of genes. So under this chapter, we will be discussing the regulation of gene expression, especially the difference of the gene expression regulation among prokaryotes and eukaryotes. Now, the lac operant and trip operant or the lactose operant and tryptophan operant, where madiscuss po ang molecular switches na mag-turn on and off sa genes. Then next is the epigenetics, the gene mutation, where dito ang madiscuss ang base pair substitution, frameshift ng mga mutation, and the evolutionary significance of the mutation. And the last topic under this chapter is the origin and transmission of genetic disorders, which will be discussed through its numerical abnormalities and structural chromosomal na abnormal activities. So now let's move now to the topic that I will be discussing, the gene regulation in prokaryotes and eukaryotes. So the intended learning outcomes of this lecture is for us to be able to describe gene regulation and to compare and contrast the gene regulation in prokaryotes and the eukaryotes. So we have here different types of cells. We have the kidney cells, the cells that we can find in our liver, in our bones, lung, brain, blood, heart, muscle, and the pancreas. Well, all of them are cells, but we all know that they have different functions. They have different shapes, no? And even though they are different from each other, they contain the same genetic bases, the same nucleotides, the base pairs, same exact sequences from one cell to another. However, these cells are different still. So now the question is, how does a cell knows which genes to express para mahimo siyang ingon ana na cell for it to be able to function in its specific pod nga trabahoon in our body. So ngano man makabalo man si cell nga dapat di ay ginanira ko ang function. No? They are all different in shape, sizes, and functions, no, but they contain the same DNA. So, same tanan to different cells has the same 3.1 billion base pairs arranged in the same exact sequences, but then why are they different from each other? It is because it is possible due to the ability of the cell to control or regulate what proteins it makes from its DNA. So si cell is na siya control to which proteins ra ang pwede nimo i-produce. No? So that's why, although there are a lot of cells, although we have the same exact sequence, but then we produce different functions, different sizes, shapes because of the different proteins that we make. So that is the concept of gene regulation. Again, gene regulation is the ability of a cell to control or regulate what proteins it makes from its DNA. So, looking here in our photo, there are two types of genes that we shall discuss. Number one is the structural genes, and the second one is your regulatory genes. So when we say structural genes, these are genes, no, those protein coding na mga genes, no? Sila ang mag-instruct sa mga structures na kinahanglan i-produce, no, sa ato ang cell. And the second type is the regulatory genes which control these structural genes na mag-produce og protein. So in our cell, there are 20,400 structural genes that codes for various proteins. However, that is only 2% of our DNA. So the rest of that, well, the 98% of those DNA that we have in our cell are all regulatory then, which is responsible for the gene regulation of our structural genes. So we all know that DNA is used to give instructions to produce proteins. However, there should be now a method of determining which genes should be turned on and off para dili ma-express tanan. Like for example, katong prognator reporter na nag-ingon na nga naman na nga to ang eyes is not producing hydrochloric acid that is being produced by our stomach cells. Because our eye cell is programmed na i-turn off to niya tong genes na makaproduce og hydrochloric acid na dapat katong nga gene is dapat ma-express siya katong sa mga cell nga nasa to ang stomach. So unsa siya dapat na unsa nga method ang makadetermine man ana nga mga panghitabo sa ato ang cell. So natay types of gene regulation that we will try to explain now, no? The positive gene regulation and the negative gene regulation. So in a positive gene regulation, the signaling molecules generate a complex that interacts with the DNA. So naatay ginatawag, no, kanina po complex is called the positive transcription factor. So this is a protein that binds to the DNA strand to increase the rate of transcription. So para mapaspas ang rate of transcription, naa si positive transcription factor na mo bind. No? And then, on the other hand, if there is a positive gene regulation para mo process ang transcription, magpadayon, mo paspas ang iyang transcription, there is a negative gene regulation in which nagayusi og signaling molecules na mag-interact with repressor proteins that dictate whether the genes will be expressed or not. No, so if natin ginatawag na repressor, no, repressor protein, na if naana siya nga mga proteins, no, it will either stop, no, the transcription or i-lesser niya ang rate of transcription. No? Ang tawag sa ilahan ng mga protein, repressor protein, otherwise called as the negative transcription factor. So again, two types of gene regulation. We have the positive and the negative, the gene regulation. So let me show you here a short video on how transcription works, no, with these different transcription factors.
By controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, A common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts nonspecifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription, or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
Okay. So in that video, no, it has been shown to us how these different transcription factor no is locating its binding site of each na mga DNA strand na iyahang gina um, adto. Okay. So again, no, not all the genes in our cell, although mo nang bahalag same tatanan og DNA no sequences bases no dili tanan na genes na nasa ato ang cell are expressed no selected lang ana nila ang ma-express no again some genes are expressed and some genes are not unang lahi-lahi pod og functions ato ang cells. So an example, no, physical example of gene regulation that happens in us humans is, for example, kanang naay infection, no, kunang mag-produce og antibody ang ato ang lawas. So, for example, that in normal, if a normal person, if walay ma-detect ang ato ang body na infection, of course, ang ato ang lawas will only produce no low rate of production sa ato ang antibody. Low rate lang kanang normal lang nga dili daghan ang iyang gina-produce na antibody. But if the cells no, in our body will detect an infection coming in our immune system, no, of course, this will trigger then and ay pa mo taas or mo increase ang rate of antibody production because naamay na sense si body na infection. So that is one way of regulation pod no nga mo function lang siya if there is something that triggers the cell itself. Another example pod is that your insulin production no, in our pancreas. So it is not all the time no na constant ang production ni insulin in our body. Magdepende siya sa glucose level sa tuang blood. So it does not produce constant rate of insulin, but it depends on the amount of glucose entering our bloodstream. So the beta cells in our pancreas then have special receptors in them, no? And that enables them to sense the amount of glucose in the blood, and we call that the glucose sensing na mga receptors. So if masense na ni glucose sensing receptors nga ang blood glucose is nagkataas, no? Ma-notice ang receptor, and then it sends the information then to the cell's DNA. And then, katuda yung mga regulatory genes nga nasa tong DNA no will respond, no, i-regulate niya nga mag-produce ang katong mga um, structural genes nga nasa to ang pancreas, no, to produce or increase the insulin secretion. Okay? Mura gud siya og kuan nga ngano mag-produce man si insulin si body kung low man ang iyahang glucose level. Katupong sa isa na to ka example, if kuan siya, mura siya og it will not benefit man the body if magsige siya og produce bisan og walay gamitan. Mo nang need na ay regulation ang atoang cell. So now, let's move forward to the main difference of the prokaryotic and the eukaryotic gene expression. So the prokaryotes regulate gene expression by controlling the amount of transcription, whereas eukaryotic control is much more complex compared to the prokaryotic gene expression. So si prokaryotic, as we all know, wala siya nucleus, si eukaryotic is naa. So in a sense of that, no, mas simpler or mas gamay lang og coverage or dili inyo na complex ang gene regulation in terms of the prokaryotic organism compared to a eukaryotic organism. So, in a prokaryotic gene expression, no, to synthesize a protein, the process of the transcription and translation occur almost simultaneously because there is a lack of nucleus within prokaryotic organism, no? So nara man siya sa cytoplasm mag-occur ang ihang transcription and translation. So most of the time they occur almost simultaneously ang iyahang processes. So si prokaryotic organism karon, if no ang iyahang resulting protein is no longer needed, for example, okay na dili na kinahanglan na magkuan ang dili na ni kinahanglan na protein, dili na siya magamit. So, automatically mo stop pod si transcription. No? And as a result of that, the primary method to control what type of protein and how much of each protein is expressed in a prokaryotic cell is the regulation of its DNA transcription. So, transcription pa lang daan. No? So, if Oh no. In prokaryotic cells, the control of gene expression is mostly at the what we call transcriptional level. No? In the transcription process, da siya ma-control in a prokaryotic cell. Now, on the other hand, in a eukaryotic gene expression, no, since naman siya mga organelles, complex manayahang structure, so the process, the transcription and translation are physically separated by the nuclear membrane. Karon, si transcription occurs within the nucleus, inside the nucleus, and your translation occurs on the outside of the nucleus, which is in the cytoplasm. So compared to your prokaryotic nga cytoplasm, both transcription and translation mag-occur. So if ang ato ang prokaryotic organism na gene expression is mag-occur lang in a transcriptional level, sa regulation in our eukaryotic gene expression, mag-occur siya no, in different levels. Natin 1, 2, 3, 4, and 5 levels na pwede siya mag-occur. Ang regulation sa gene, pwede siya ma-change or ma-control. So it could be controlled or regulated when our DNA is uncoiled and loosened from the nucleosomes to bind transcription factors, or what we call the epigenetic level. Then another is when the RNA is transcribed or in the transcriptional level, and it could be regulated when the RNA is processed and exported to the cytoplasm after it is transcribed or the post-transcriptional level. It could also be regulated when the RNA is translated into protein or in the translational level, or after the protein has been made or in its post-translational level. So several levels of the DNA expression to it into protein, no, pwede siya ma-regulate, no, depending on the processes that will happen in each level. No? So in here, for example, na tayo ginatawag ng ang regulation may occur when DNA is uncoiled and loosened from nucleus onto by transcription factors. So, kung ang ato ang what do you call that? Ato ang DNA is coiled siya in a histone, so dili siya ubanan sa iyang transcription factor, so dili siya maka-follow sa transcription. So ang kato nga mga what do you call, the DNA strands nga naa sa histone, bounded with the histone, is silent sila. So, yun, genes bounded histones can't be expressed. Sa silence man sila, kay naman sila sa histones. So, there is a way, no, a way to express those genes nga na siya sa histones para ma-unbound sila diha is when, no, ang ato ang histone will undergo acetylation, methylation, and phosphorylation, no, processes para ma-disbound sila within that histone kanang protein, and then pwede na nga mo more siya mo adto sa ilaha ang katong mga transcription factor para masugdan na og process. So in this figure, no, is a illustration of those what we have described earlier. The prokaryotic here in figure 1, letter A, now the prokaryotic transcription and translation that occurs within the cytoplasm of the prokaryotic organism. And then in letter B, the eukaryotic gene expression, now that is regulated, no, in the transcription process and RNA processing within the nucleus, and the protein translation in the cytoplasm. So here is a table of a summary on the differences in the regulation of gene expression of prokaryotic and eukaryotic organism. So in your prokaryotic, walay nucleus, eukaryotic contains nucleus. In prokaryotic, DNA is found in the cytoplasm, whereas in the eukaryotic, the DNA is confined to the nuclear compartment inside the nucleus. In the prokaryotic organism, the RNA transcription and protein formation occurs almost simultaneously. And in your eukaryotic organism, the RNA transcription occurs prior to protein formation. Bago na ang transcription, then later on protein formation. And then it takes place in the nucleus. Then the translation of RNA to protein occurs inside the cytoplasm. Then the last key point is that the gene expression in the prokaryotic organism is regulated primarily at the transcriptional level. And in the eukaryotic organism, the gene expression is regulated at many levels. It could be in the epigenetic level, it is transcriptional during transcription, post-transcriptional, translational, and post-translational level. So, pwede siya ma-regulate in various na levels. So, in the next lesson, you will be learning on the molecular switches that turn gene off and off. No, kaya tayo ginaingon na kung sa may maka-on and off ato nga mga genes nga nang dili man ma-expressan aside ato mga histones that allow gene silencing. So, sa pa may other switches, no, kan unsa nang masugatan nato in the next lesson ang mga term ng mga operon, mga repressor, mga C-amp, and other mga activators, enhancers na mga terminologies. No? Ba-discuss dira apod in the next lesson po ang mga lock operon and trip operon, mga theory first, mga theory na siya una, then gi studyhan with our scientists no on how these processes um regulate no the genes of I think bacterial cells ni sila operon, trip operon. So that's it. No, that ends my um recorded report lecture now for this topic. And I hope you have learned something from that. And thank you for listening, and God bless us all.